Hi everyone and welcome back again to Biology Circuit. In this video, I am going to discuss some important questions from methods in biology sections of CSIR NET. This is the last unit of CSIR NET Life Science syllabus, and every year lots of questions comes from this unit. So let's start. First question. Which one of the following techniques will you use to identify more than 1000 differentially expressed genes in normal and tumor tissues in one single experiment? So the option is RAPT, genome sequencing, chip assay and transcriptome analysis. So the right answer will be the transcriptome analysis. So here you can see that in the question it is mentioned that to identify more than 1000 differentially expressed genes. So in that case, transcriptome analysis, that is kind of RNA analysis actually, RNA sequencing technique. So option 4 is the right answer. Which isotype below is best suited for metabolic leveling of glycerolide 3-phosphorehydrogenase? So the right answer will be option 1. C14. The next question. For identification of three proteins moving together upon loading in a single lane of a SDS paid gel, the best method is, the right answer will be option 3. Western blot followed by stripping and reprobing. Next. Which among the following is the simplest method to estimate the concentration of glycerol in an aqueous solution of glycerol? Option 1 is UV absorption spectroscopy, option 2 gas chromatography, option 3 pH measurement and option 4 viscosity measurement. So in this case the right answer will be the option 4, viscosity measurement. It is the simplest method to estimate the concentration of glycerol in an aqueous solution of glycerol. Normally glycerol also viscous in nature, so it is quite easy to find out this answer. Which one of the method listed below is the most sensitive level-free quantification method for proteins? UV spectroscopy, infrared spectroscopy, Raman spectroscopy, C13 content of protein. So the question is level-free quantification method of proteins. The right answer will be option 1, UV spectroscopy. Next. A researcher would like to monitor changes in the level of serum protein for which an antibody is available. Which one of the following methods would be best suited for the purpose? Immunofluorescence microscopy, fluorescence in situ hybridizations, enzyme-linked immunosorbent assay, or fluorescence activated cell sorting. The right answer will be option 3, enzyme-linked immunosorbent assay. That is ELISA technique. ELISA is a very important technique in immunology. Next. From the various techniques listed below, which one cannot be used precisely map the transcription start site of a gene? Okay. So the option is S1 mapping, option 2 sequencing the region downstream of promoter, option 3 5' base, RACE, and option 4 primer extension method. So here the right answer will be sequencing the region downstream of promoter. That cannot be used precisely to map the transcription start site of a gene. Okay, because the transcription start site, it could be upstream. Next question. Which one of the following assay systems can specifically detect apoptotic cells? Option 1, tetrajulium dye based colorimetric assay, that is MTT assay. Option 2, FITC annexin 5 based FACS analysis. Option 3, 5-on Csir release assay. Option 4, trypan blue exclusion assay. So option 2 is the right answer. FITC annexin 5 based FACS analysis can be specifically detect apoptotic cells. Next. Protein conformation dynamics cannot be determined by which one of the following technique? NMR spectroscopy, differential scanning calorimetry, mass spectroscopy, fluorescence microscopy. So the right answer will be option 4, fluorescence microscopy. The question is protein conformational dynamics, which cannot be determined. So NMR spectroscopy, differential scanning calorimetry, mass spectroscopy, all these three techniques can determine. By using these three techniques, you can determine the protein conformation dynamics. Fluorescence microscopy is generally used in the molecular biology and cell biology technique. Next, the presence and distribution of specific mRNAs within a cell can be detected by northern blot analysis, RNase protection assay, in situ hybridization, real-time PCR. So the question is related to the mRNAs. So the right answer will be the northern blot analysis. So in case of DNA, it will be the southern blot analysis. Okay. So next, the bioinformatics tool used to find the sequence similarity in the subunit of hemoglobin is. The right answer will be option 3, HAMR. Which one of the following DNA markers can be used to distinguish between homozygote and a heterozygote? RAPT, AFLP, RFLP, ISSR. So the right answer will be RFLP. RFLP can be used, or is a DNA marker that can be used to distinguish between homozygote and heterozygote. Next. Orientation of a closed DNA fragment in a plasmid vector can be checked by PCR using 2 gene-specific primers, restriction digestion with the enzyme that has a single restriction site within the clone gene and none in the vector. Option 3, PCR using a combination of 1 gene-specific primer and 1 vector-specific primer. Option 4, restriction digestion with the enzyme that has 2 restriction sites within the vector sequence and none in the clone gene. Here the right answer will be option 3. PCR using a combination of 1 gene-specific primer and 1 vector-specific primer. So the orientation of a closed DNA fragment in a plasmid vector can be checked by PCR technique using a combination of 1 gene-specific and 1 vector-specific primer. Next, the multi-mass spectrum of a peptide give a single peak width by set of 2000. The ESI mass spectrum of the same peptide gave multiple peaks. The observation indicate that. So this question is from mass spectrometry. The right answer will be the option 2. Multiple charged species of the same compound are observed in the ESI spectrum. So multiple charged species of the same compound are observed in the ESI spectrum. That's why you get it gives a multiple peaks for the same peptide. Okay. Next. A multimeric protein, when run on SDS gel, showed 2 bands at 20 kDa and 40 kDa. However, when the protein was run on a native gel, it showed a single band at 120 kD. The native form of the protein would be, obviously it will be heterotetramer. Okay. Because when it run in the SDS gel, it shows two bands of 20 kD and two band at 40 kD. So the combination will be, and the total molecular weight is 120 kD. So 40 plus 40, 80, and 20 plus 20, 40. 80 plus 40, that is 120. So the protein is actually a heterotetramer. Which one of the following combinations of terms is matched incorrectly? Nanopore, DNA sequencing, right? Pyrosequencing, protein primary structure. No, it is wrong. Pyrosequencing will be also kind of DNA sequencing method. Homologous recombination, chlorophos transformation, and option 4, SSR, codominant markers SSRs. Okay, so the incorrect option is pyrosequencing, protein primary structure. So the right answer will be option 2. Which one of the following options represent a combination of terms that are matched incorrectly? DDNTP's chain termination, right? Southwestern blot, physical interaction between DNA and proteins. Okay. 5' to 3' exonuclease activity, proofreading polymerase for PCR. That is wrong. And this two hybrid system, interaction between proteins. So option 1, 2, 4 are correctly matched. So the incorrectly matched option is the option 3. Option 3 is the right answer. Fluorescence microscope that require photoactivatable probes to obtain super resolution is, the right answer is option 2, T-STOM, stochastic optical reconstruction microscope. Answer 2. To examine in vivo colocalization pattern of two different proteins using fluorescently labeled antibodies, which one of the following combination of fluorochromes will be appropriate? The right answer will be option 1, Alexa 488 and Cy5. So it can be used to examine the in vivo colocalization pattern of two different proteins using fluorescently labeled antibodies. Next, which one of the following factors will not have any impact on the resolving power of a bright field microscope? The color of light, intensity of light, angle of admittance of light in the objective lens, and option 4 is medium between objective lens and specimen. The right answer will be intensity of light. So intensity of light will not have any impact on the resolving power of a bright field microscope, and the other three options has some impact on the resolving power of a bright field microscope. So that's all for today. Hope it will be helpful, and if you like this video, please subscribe to Biology Circuit and share it, and thanks for watching.